Hi, my name's Sarina Mehta, and I'm a junior at Scarsdale High School. My Polygence project analyzes the link between Alzheimer's disease and traumatic brain injuries. Neurological disorders are currently affecting 15% of the worldwide population, are the second highest cause of death, and the leading cause of disability worldwide. A study by Global Burden of Disease explains that the number of people living with brain disease is expected to double by 2050. And within the wide range of neurological disorders, Alzheimer's disease and Parkinson's disease are the most common neurodegenerative diseases. But for today's discussion, we'll be focusing on Alzheimer's disease. Alzheimer's disease is a neurological disorder that impairs a person's memory and other cognitive abilities. The chart on the right shows some of the symptoms such as memory loss, difficulty in decision-making and judgment, difficulty in problem solving, confusion, and mood swings. Currently, 6.2 million people are suffering from Alzheimer's disease in the US, and that number is expected to go up to 14 to 16 million by 2050. Common causes of Alzheimer's disease have been identified as aging, genetic propensities, environmental and lifestyle factors, and traumatic brain injuries, which we are investigating today. The CDC defines traumatic brain injuries as a disruption in the normal function of the brain that a bump or blow can cause, a jolt to the head, or a penetrating head injury. Seen on the chart, TBIs can occur from falls, motor vehicle crashes, and assaults, but also suicides and sport injuries. But falls are the leading causes of TBIs among the elderly. They can be categorized as mild, moderate, or severe based on the type of injury, location in the brain, and age and gender of the individual. While most moderate TBIs can cause loss of or deceased consciousness, amnesia, muscle weakness, loss of vision, change in speech, disorientation, slow thinking, or difficulty in concentration, severe cases result in extended periods of unconsciousness, coma, or death. Growing scientific evidence has demonstrated that a TBI can result in many pathologic events, like neuroinflammation, oxidative stress, cerebral vascular impairment, also known as edema, circulatory insufficiency, and a blood-brain barrier breakdown. Each of these events can contribute to long-term cognitive and emotional impairments in TBI patients. TBIs can penetrate the blood-brain barrier, which regulates the transfer of solutes and chemicals between the circulatory system and central nervous system. It protects the brain from toxins that may be present in our blood while allowing nutrients to reach the brain. The precise control of the central nervous system, homeostasis, allows for proper neural function while protecting the neural tissue from toxins and pathogens. Alterations of these barrier properties are an important component of the pathology and progression of different neurological diseases. Now let's look at the physiology of Alzheimer's. We know neurons carry messages from the brain to the muscles and organs of the body. Any toxic changes in the brain that hinder this process leads to neurological disorders. Alzheimer's disease develops from necroptosis, or cell death, of neurons, which is caused by accumulation of amyloid plaques and neurofibrillary or tau tangles. As you can see in the diagram on the right, amyloid plaques are beta-amyloid proteins found in the tissue between neurons, and neurofibrillary tangles are found within the neuron and are formed by the hyperphosphorylation of a protein called tau. In healthy neurons, the tau proteins support the function of the microtubules to deliver substances throughout the neuron. When the tau proteins twist into tangles and aggregate in an insoluble form, they interfere with the transport system of the microtubules, which hinders communication between the neurons and leads to necroptosis. To prevent the accumulation of beta-amyloid proteins, the apolipoprotein E clears the buildup of AB plaques with proteolytic degradation, or the breakdown of proteins into smaller polypeptides or its respective amino acids. In healthy brains, the protein fragments, or any foreign substances or debris, are broken down and eliminated by microglia cells. But in Alzheimer's brain, the microglia cells fail to eliminate the overabundance of AB fragments. Unfortunately, not all strands of the apolipo-E protein are effective in removing AB plaques. The main strands of apolipoprotein are apolipoprotein E2, apolipoprotein E3, and apolipoprotein E4. Apolipoprotein E4 induces neuronal loss, age-related cognitive decline, and neurotoxicity when synthesized in neurons due to the arginine 112, leading to domain interaction and the structural alterations. Apolipo E4 fragments, resulting from neuron-specific proteolysis, escape the secretory pathway, causing detrimental effects like enhanced tau phosphorylation, neurofibrillary tangle formation, cytoskeleton alteration, mitochondrial dysfunction, and abnormalities in the mitochondria-associated membrane. The definitive link between TBIs and Alzheimer's like neurodegeneration is not known, but the pathogenesis of TBIs and AD exhibits similar aberrations. Early stages of both Alzheimer's and TBIs involve lipid metabolic alterations, cell membrane damage, and organelle dysfunction. Cardinal AD features such as AB disposition, tau hyperphosphorylation, and neurofunctional deficits are reported in TBI models. Key studies have also shown that adults with a history of moderate TBI have a 2.3 times greater risk of developing Alzheimer's, and those with a history of severe TBI have a 4.5 times greater risk. While single TBI episodes often result in full recovery, a history of multiple TBIs raises the risk of developing Alzheimer's. A study by Agrawal et al. Utilized the controlled cortical impact model of a traumatic brain injury in adult mice to observe an increased functionality of mitochondria-associated endoplasmic reticulum domains after a single moderate injury. Mitochondria-associated endoplasmic reticulum is a conduit that allows communication between the endoplasmic reticulum and mitochondria. An increase in mitochondria-associated endoplasmic reticulums may promote mitochondrial AB deposition. To administer the study, a controlled cortical impact injury of moderate severity was induced on adult male mice, penetrating the cortex. Molecular pathways were analyzed 1, 3, and 7 days after injury and compared to naive or uninjured tissue samples. The study observed a controlled cortical impact model in adult mice showing increased functionality of mitochondria-associated endoplasmic reticulum domains after a single moderate traumatic brain injury, similar to Alzheimer's disease. Evidence of this was attributed to elevations in C9-9 after TBI showed an increased localization to mitochondria-associated endoplasmic reticulum domains after 3 days of injury. An increase in C9-9 levels were driven by increased cholesterol transporter APOE, lipid transporter LDLR, and lipid efflux pump ABCA1, suggesting a mitochondria-associated endoplasmic reticulum upregulation. Mitochondria-associated endoplasmic reticulum plays a crucial role in lipid metabolic disturbances post-brain injury, suggesting traumatic brain injuries as a potential factor that contributes to the development of Alzheimer's. The data also supports an active role of C9-9 and cholesterol trafficking in triggering mitochondria-associated endoplasmic reticulum activity after a TBI. Thus, the study supports the hypothesis that traumatic brain injury episodes have the potential to induce C9-9-mediated upregulation of mitochondria-associated endoplasmic reticulum, disturbing lipid homeostasis, resembling Alzheimer's molecular and cellular phenotypes. The study by Mielke et al. Seeks to understand the link between traumatic brain injuries and Alzheimer's by using a population of patients with a medical record of traumatic brain injuries and Alzheimer's disease-related dementia. The study identified patients with a diagnostic code indicative of a potential traumatic brain injury, which has over 45,000 patients. From this population, the study further identified patients who were over 40 years of age and had at least 5 years of follow-up with a confirmed traumatic brain injury. For the 1,418 patients who met the criteria, 2,836 individuals were identified as age and sex and non-head trauma reference. The study showed that exposure to any severity of traumatic brain injury at age 40 and older was associated with an increased risk of developing Alzheimer's disease related dementia by 1.3-fold. However, it did not find a link between traumatic brain injuries and specific dementia types, such as Alzheimer's disease. Malike et al. Contend that other studies with larger sample sizes and Alzheimer's disease-related dementia outcomes also did not find any association. Additional research is necessary to determine the relationship between traumatic brain injuries and Alzheimer's disease-related dementia. The third review by Ramos Cejudo evaluates the connection between AB and tau deposition after TBIs and the development of neurodegenerative diseases like Alzheimer's. The review postulates that the cerebrovascular dysfunction is an important contributor to neurodegeneration after a traumatic brain injury. In this study, Ramos Cejudo explored both animal and human studies with a focus on cerebrovascular dysfunction. They're contributing to the understanding of the consequences of traumatic brain injuries on the development of Alzheimer's-like pathology. By exploring and compiling findings of many animal and human studies, the review found that the accumulation of AB proteins and tau tangles have been found in post-mortem brains of people with TBIs or people subject to repeated concussive head injuries. They also found that TBIs can induce acute blood-brain barrier disruption, which results in accumulation of AB protein. Studies in animal models show that injury causes tau to become phosphorylated, misfolded, aggregated, and cleaved. The experimental data shows that AB and tau releases from a TBI lead to cerebrovascular injury. However, the cerebrovascular injury further induces AB and tau deposition, creating a feed-forward loop that can ultimately lead to the development of AD-like pathology. Studies of transgenic mice showed tau overexpression can initiate blood-brain barrier breakdown in vivo, suggesting that tau may play a role in blood-brain barrier dysfunction. Study shows that any alteration in brain waste clearance after a TBI is responsible for AB and tau fragment accumulation. Although further studies need to be conducted to understand the many confounding factors that may contribute to TBI-induced impairment of neurovascular function to AD-like pathology. While there are currently no specific treatments approved specifically for TBI-induced Alzheimer's disease, individuals who have experienced a TBI are at risk for or have developed Alzheimer's disease can seek out treatment plans aimed to manage their symptoms. Currently, the first line of defense for Alzheimer's patients includes acetylcholinesterase inhibitors such as rivastigmine, galantamine, donepezil, and N-methyl D-aspartate receptor antagonist, more commonly known as menantine. While these treatments curb symptoms, they are not able to prevent the progression. On the other hand, immunotherapy, or utilizing the patient's immune system to target abnormalities, is gaining attention as it has been known to slow the progression of the disease. There are two types of immunization: active immunization and passive immunization. Active immunization is a vaccination where a fragment of AB is administered to stimulate an antibody response. However, it is not approved by the FDA. Passive immunization uses monoclonal antibodies, which are synthetic peptides, to reduce the amount of AB in the brain by triggering the immune system to break down the AB proteins. In 2023, the FDA approved a passive immunization known as Leqembi. It is the first amyloid beta-directed antibody that reduces AB plaques that form in the brain. Lecanemab is given as an IV infusion every 2 weeks and has been shown to slow the progression of AD by 27%. The presence of amyloid plaques and tau proteins are key features in the pathology of both TBIs and AD. They suggest a potential link between the two conditions. And studies have indicated that individuals with a history of moderate or severe TBIs face a substantially higher risk of developing Alzheimer's. However, a conclusive link has yet to be established. To further explore the link between TBIs and Alzheimer's, 3 reviews were cited. The first showed single moderation TBI induced disruptions in MAM regulation in mice, leading to alterations in lipid homeostasis across different cell populations in the brain. The second investigated TBI as a potential risk factor for developing ADRD, as it found that exposure to any severity of TBI aged 40 or less was associated with a 1.3-fold increase. They also found that there is no link between AD and TBI, but TBIs are a risk factor for ADRD. The last study highlighted cerebrovascular consequences, including blood-brain barrier disruption and alterations in cerebral blood flow, initiating a feed-forward loop involving AB and tau deposition, ultimately leading to an Alzheimer's-like pathology. As there is still no cure for Alzheimer's, the main treatments, such as acetylcholinesterase inhibitors, NMDA receptor antagonists, and immunotherapy, are therapeutic and mainly focus on managing symptoms which do not change the underlying disease pathology. As a first step towards addressing cardinal features of Alzheimer's, one form of immunotherapy has been approved by the FDA in 2023. Lecanemab is the first amyloid beta-directed antibody that reduces AB plaques that form in the brain. Annual federal funding for Alzheimer's research was $3.7 billion in 2023, up from $504 million 10 years ago. I'm hopeful this unprecedented level of funding will allow scientists to enhance disease knowledge, identify new biomarkers for early detection, and develop effective drug treatments for those at risk or affected by Alzheimer's. And lastly, thank you, Polygence, for this amazing research opportunity. Most of all, I really appreciate the time my mentor Alejandro has dedicated to allow me to investigate this increasingly important topic. Thank you.